Today I'm going to share with you some background information about this manuscript, Evolution of a Polyphenism by Genetic Accommodation. So first, we need to get rid of some vocabulary terms and define some of these terms. So phenotypic plasticity is not a complicated concept, but it is sort of intellectually and genetically difficult to understand sometimes. But this is the ability, as it says there from Wikipedia, for an organism, say an organism with the same genotype, to have different phenotypes or traits depending on its environment. So this example here shows different grasshoppers from different environments showing different coloration traits or sizes. And those traits and sizes are independent of genetics. These are all genetically identical, essentially organisms, but their phenotypes or traits change based on the environment. And so here are some examples of what phenotypic plasticity can include, like morphology, body shape and size, physiology. An example of this is that organisms can acclimatize to different or acclimate to different environments. So what may be initially cold to you might not feel as cold over time. You might get used to that environment. And that can involve physiological changes within an organism that correspond to a change in the environment, and also behavior. It's really important to point out that plasticity happens within the lifetime of an individual, not across generations. So this is not Lamarckian evolution. Lamarck had this idea, which is illustrated here, that if a food source got higher and higher, for example, in Africa where there are giraffes, if the food got slightly higher every generation over time, then the individuals would have their necks grow generation after generation. The necks would get longer to keep up with this environmental change. And we know that that's not how evolution works. Evolution isn't Lamarckian. It's not something about change based on the environment that doesn't have to do with DNA. Lamarck was just suggesting that the giraffe physically stretches their neck, and that physical stretching somehow passes that trait on to the next generation. We know that inheritance is based on the passing of DNA from generation to generation. And we know that in this sort of an example, the cause of the inheritance of a longer neck, if this were the case in giraffes, would be because there were mutations in developmental genes, probably, that enabled all of the offspring of that longer necked ancestor to also have longer necks. So again, polyphenism, phenotypic plasticity, we'll get to polyphenisms in just a second. Phenotypic plasticity means within the lifetime of an organism, from early development to adulthood, the environment affects that individual's traits. So polyphenism. The two roots of this word, poly and phenism, are essentially many and traits or phenotypes. So an individual is polyphenic, or a species is polyphenic, if a single individual can either adopt one of two or more different traits depending on their environment. So in other words, here are genetically identical dung beetles, but they have a polyphenism. They have two different, despite the fact that these dung beetles have the same DNA, therefore the ability to make exactly the same body structures, they don't actually make the same length horns because of conditions. In this case, as it says here, it's depending on the amount and quality of dung provided by the mother when these two individuals were larvae. So that is an environmental condition, the quality and the amount of the food that these two beetles have early in life affect a trait later in their own life. So again, not Lamarckian. This isn't change in the next generation. This is a difference between individuals of the same generation depending on environment. So polyphenism is a form of phenotypic plasticity. And what this means, phenotypic plasticity, is trait flexibility, the ability for a trait or the existence of a trait that can have different forms. And an example of this are pea aphids, which, depending on the season environment, will either have asexual reproduction or sexual reproduction. This is the same species. These are all genetically identical individuals, but the environment, in this case season, somehow dictates whether or not the individuals that are produced will be asexually reproducing hermaphrodites, or if they will then produce sexually reproducing males and females. So polyphenism, the ability of genetically identical individuals of the same species to have one or of two or one of more different possible traits depending on an environmental condition. And so those sorts of traits are considered plastic. They're not rigid. They're able to be molded. One way to look at this is to consider norm of reaction plots. And this is a way to tell, and this is a way that geneticists and evolutionary biologists use to know if, or to measure, the amount of plasticity of a trait. So if you consider that there could be three different genotypes of an individual, one genotype that's a red genotype, a different genotype that's a green genotype, and a third genotype that's a blue genotype. Now consider what happens to those three different genotypes in an environment. So the x-axis here indicates that the environment is changing. So each point on this x-axis would indicate a change in some sort of environmental condition. Imagine temperature, low temperature, high temperature. If each of those three genotypes in the same species has the same trait, regardless of the environment, that indicates that that trait is not plastic. It doesn't change based on the environment. A trait that's plastic will show that depending on the genotype, the trait will change as the environment changes. So that's the definition of a plastic trait. And then this third example is a very extreme example, which involves what they call strong genotype by environment interactions, which indicates that while some genotypes might have a change in one direction in increasing temperature in this example, other genotypes might have opposite reactions to the same environmental change. And these colored lines are referred to as reaction norms. They're plots of genotype and phenotype to indicate whether or not individuals with the same genotype have changes in the trait depending on that environmental condition or variable. And that again is how we define a trait that's plastic. And an example of a non-plastic trait is homeostasis, any mechanism that is meant to keep the trait the same regardless of the environment. So that, for example, is endotherms. Endotherms are able to maintain body temperature despite what's going on outdoors. So all of this is able to occur, especially homeostasis, because of what's called evolutionary capacitance. So I'll read the blue examples for you here, that these mechanisms, whatever proteins and mechanisms we have that facilitate homeostasis, basically what they do is they suppress any mutations or genetic variants, same thing, that could cause homeostasis to be disrupted. And interestingly, for evo-devo, it's the things that disrupt homeostasis that can often lead to the evolution of developmental change. Remember, development is meant to limit the variability and to produce the same sorts of organisms faithfully over and over again based on the DNA sequence in that organism's nucleus. So we have these homeostatic mechanisms, and they, by suppressing any genetic variation that might cause changes in development, they act as a sort of a capacitor. So you can imagine that over generations, an individual, and I'll diagram this in just a minute, over generations, an individual might have some mutations occur that could make slight changes to the phenotypes, the traits of that lineage. But they don't because of homeostatic mechanisms that suppress the effects of those mutations. And at some point in time, and that's what capacitance is referring to here, at some point, maybe there's the last mutation that is what we'd say is the straw that breaks the camel's back. The individual has sustained too many mutations, and all of a sudden, all of those independent mutations that have been accumulating over time are finally all revealed at once. The ability for homeostasis to suppress them is overwhelmed. And all of a sudden, in one generation, after that last mutation, there's a huge explosion in phenotypic change, not a true explosion. All of that phenotypic change is revealed suddenly. And that can look like an individual that's an offspring looks totally different or acts totally different. It has some phenotype that's very different from the parents in a surprising sort of way. That's the idea of capacitance. And so an example of capacitance is as chaperone proteins. So we all produce heat shock proteins. These are proteins that are encoded by genes in our genome, and many species have these, not just humans. And their role is, as new proteins are being translated and produced, these proteins help fold those new proteins, so heat shock proteins help fold the new proteins into the correct shape, the shape that's intended for those proteins to be in to function properly in the cell. And what happens in heat stress is that those correctly folded proteins can become unfolded or misfolded. And those heat shock proteins also act in this process to refold denatured proteins or unfolded proteins, to refold them into the correct shape. So for decades now, chaperone proteins, heat shock proteins, have been really sort of highly regarded as a great example of both a homeostatic mechanism. They prevent change, in this case based on misfolding of proteins. They also then can act as a capacitor. So for example, to make that point, imagine you've got a number of mutations in the genome that make those proteins misfold. So let's say there are a couple of chromosomes and there are some genes that produce some proteins, and those, let's just say a couple of them, have mutations in them so that let me make this diploid. So we have heterozygosity for these. So these two genes with mutations, there's one mutant form, one wild type form, one mutant form, one wild type form. So let's say the wild type form here is meant to be a circle, and when you get the mutation, that makes a circle with a divot in the protein. So it folds differently based on that mutation. And let's say the same is true over here. The wild type copy of the gene is a square or a box, and the mutant version is shaped like a triangle. Just making stuff up. So in the presence of chaperone proteins or heat shock proteins might bind to those misfolded proteins. So in this case, the circles are the heat shock proteins. And they could cause them to fold back into their correct shapes. Then the question is, what happens if there are a lot of these and all of a sudden the gene that encodes the heat shock protein gets a mutation? So now those heat shock proteins aren't functional as heat shock proteins. So imagine the slide we just went over being a previous generation. And now for the first time, we have this new mutation that makes the heat shock proteins dysfunctional. It makes them shaped like, I don't know, two circles. Figure eight. Now, when you have that previous situation where you have a couple of genes and one of them is mutant and another chromosome, there were another pair of genes, one of them was mutant, this heat shock protein no longer can function to refold those mutant proteins. That is a form of capacitance, is that all of a sudden, this chaperone protein, chaperone gene mutation, all of a sudden lets all of these different mutations in protein coding genes be revealed in a single generation when that mutation makes the heat shock protein, the capacitor, stop working. And all of a sudden, all of this variation based on the traits conferred by these mutant genes, like this one and this one, are revealed. So one generation, you can go from parental sort of wild type or normal phenotype to an offspring if they have this mutation that can look or act or behave or have different traits, very different traits potentially from their parents. And so this is a great quote from one of the authors in a news article about the manuscript that we're reading. And so I'll let you read it. I highlighted the blue parts particularly for this case. Again, a capacitor in electronic terminology is an element that stores up charge and then releases it suddenly. So as an example of that, let's think about a situation where we have time plotted on the x-axis. That could be in generations, for example. And we could have two different y-axes. We could have genetic change, the number of mutations. And then on the other y-axis, we could have phenotypic change or trait change. So under capacitance, you might see mutations occur slowly over time. Every generation, there might be one or two new mutations. So that over time, much and greater amounts of genetic change have occurred from generation to generation. But you might not see any phenotypic change. The phenotype, whatever that phenotype is, stays constant. So there's no phenotypic change despite the fact that genetic change is occurring. This is what, for example, those heat shock proteins might facilitate. Slow and steady random changes in coding regions by mutations, but the trait stays the same. And then in the same sort of graph, we could have a situation where, like that mutation in a capacitor gene, like a heat shock protein, let's redraw that because genetic change will never go down, which I just accidentally drew. Get rid of that. If one of those mutations happens to be in, for example, the heat shock protein, then even though you would see very little phenotypic change over time, all of a sudden when that last mutation happened that was really important for suppressing the effects, the phenotypic outcomes of all those mutations, all of a sudden you'd see a jump in the trait associated with when that mutation occurs. It doesn't look quite like that, but there we go. So the mutation happens there, the phenotypic change happens there, and then depending on what happens next, if there are other capacitors, for example, that trait might, again, remain steady over time despite the fact that there are an increasing number of mutations again. Or I suppose it's also possible that at that point, each one of those little genetic changes would also cause a corresponding subtle but visible, I suppose, change in the phenotype. So that's an example of capacitance, and then what happens when capacitance disappears. So a major question in evo-devo then is what sorts of genes and processes and proteins can be those sorts of capacitors that allow genetic changes to build up slowly over time and not reveal changes in traits, but then suddenly, given some sort of an environmental change, or in this case, a protein change, but an environmental change might do this too. It doesn't just have to be a mutation in a heat shock protein. This could be, for example, global warming, that all of a sudden there be not a genetic change, but a change in the environment, right? we talked about environmental effects on traits earlier, plasticity. So instead of genetic change, this axis could be environmental change. And there could be slow environmental change over time, but all of a sudden, say temperature might cross a threshold where the trait suddenly changes. And that's what we're going to be looking at in this paper. So the goal of this paper is to understand how plasticity works and approach this by trying to evolve polyphenism. That is, to make an organism that is not polyphenic, doesn't have varying traits depending on environment, have a trait like that. And they're going to do this based on environment, and they're going to use the tobacco hornworm, Manduca sexta. So the general idea here is the authors think what would be ideal is you find two different species that are closely related, so sister species. When I say a pair of species, really I mean very closely related species, where one of them has a polyphenism and the other doesn't. And the idea here is that if one species has a polyphenism, that polyphenism is probably based on some genetic architecture basis that the sister species, the other species that isn't polyphenic, doesn't quite have the same genetic architecture for. So it's maybe close, the sister species is maybe close to becoming polyphenic, but it maybe needs one or two more mutations to get it to the point where the environment can cause an effect on the trait. So I should also mention this idea of canalization of a trait. So canalization is not a difficult concept. It's kind of hard to explain again, like all of the other traits or terms we've been talking about so far, polyphenism, plasticity. Canalization, I guess you could kind of think about as a canal, a ditch in the ground that's meant to carry some sort of liquid like water. And if you have a shallow canal, that means it's easy for maybe if there was an earthquake or a heavy truck rolled by, some of the water to slosh out. And that would mean that this water is not very canalized. It's easy for it to change or to move. And when we talk in developmental biology about canalized traits, we're talking about traits that are robust in the face of, say, environmental change, like an earthquake or a truck driving by. So a canalized trait resists change. The barriers of the canal, the walls of the canal, are maybe you could consider as the genetic architecture of the organism and its capacitors, the heat shock proteins. So maybe this organism or this trait has lots of different capacitors that help maintain homeostasis, that buffer change. So it would be hard, really hard, it would take a lot of energy or mutations to slosh that water out of that canal. So that would be a deeply canalized trait. Traits that aren't very canalized maybe don't have a lot of protection against change. Not so many homeostatic mechanisms that try to buffer them against change. So the point here is that if there are two species and they're closely related and one has a polyphenism and the other doesn't, that probably means that the canalization isn't really deep. It's evolutionary, this trait is evolutionarily recent, so there might not have been a lot of time for that trait to become deeply canalized, for lots of supporting homeostatic mechanisms to have evolved to prevent change. So in this study, they use these two different species of Manduca, Manduca sexta and Manduca quinque maculata. You can just call them sexta and Q if you want to. And one of these species, as we just discussed, has a polyphenism rather, the other one doesn't. So Manduca sexta, the tobacco hornworm, is green if it grows at 20 degrees or 28 degrees. So the environmental change we're going to talk about here is temperature change. And this is not polyphenic, this is monophenic. That suggests this trait is canalized. They're green regardless of what temperature they're grown at. So in that environmental change on that axis, This sort of temperature change doesn't change the phenotype, but a closely related species in the same genus, Manduca, is polyphenic. Its trait is not canalized. The trait can change depending on the temperature. So these are black at 20 degrees and green at 28. So that trait is the same in Manduca sexta and MQ, but here is that polyphenism, that in a single species, depending on the environment, there can be a change in trait. Again, this isn't a change in one individual's lifetime. This is a change between individuals depending on the temperature that they develop in. So one individual that develops at 20 degrees would become black. If you took that exact same individual, or a twin of that individual, for example, and instead had reared it at 28 degrees, that same genotype of individual would turn green, would develop as a green Manduca. So that's a polyphenic trait. A really critical part of this study is that the authors have a mutant version, of Manduca sexta called the black mutant. And it's called the black mutant because it isn't green at 20 degrees, which is a typical rearing temperature. They're black, like Manduca quinque maculata. And the mutation, though, is that not only are they black at 20, but they're also black at 28 degrees. The cool thing about this, and this is the mutant that the authors are going to use in this paper, is that this is polyphenic, but it's not apparent based on that initial rearing temperature. A constant 20 degrees will produce a green Manduca sexta, and a constant 28 degrees will produce a green Manduca sexta. And a constant growth at 20 degrees in the black mutant makes black, not green. But if you heat shock either one of these, actually should have put the asterisk on both of those. If you heat shock a black mutant, heat shock it, not grow it at a particular temperature. 28 degrees is hotter than 20. We're talking about a hotter, brief pulse of heat shock early in development. Those individuals don't develop as black. They develop as various shades of green, as explained here. So this suggests, well, I'll tell you that in a second. Let's get to the bottom of the slide. So the question is, can the authors evolve this black mutant to, instead of being black at 20 and 28 degrees, can they evolve it to be consistently green when grown at 28 degrees, not just sometimes green when they're heat shocked and grown at 28 degrees? Oh, I should go back to that slide and make one other point. And that is that this variation that we see in the black mutants, in some individuals can become very green when they're heat shocked, some less so, some barely green. So there's a lot of variation in the phenotype when these mutants are heat shocked. And the variation might mean that there's genetic variation among individual Manduca sextas that can cause them to respond differently to heat shock. So for example, some individuals when they're heat shocked, if they turn really green after heat shock, that might mean that their genomes have some capacity to override the black mutation that causes them to develop black pigment instead of green pigment. And some other individuals, when they're heat shocked and produce offspring, and those offspring develop, maybe they become only a little bit green because the genotype of their parents only allowed them to produce a little bit of green pigment as opposed to a lot in some of the other mutants. So again, the idea here is that it's possible that the reason that there's variation in the amount of green produced after heat shock in these mutant worms is because there could be some genetic variation among the parents that produce those larvae that are heat shocked and grow up to be different colors. So the way that the authors are going to approach this experimental evolution is they're going to try to force the green phenotype that's produced when they heat shock these black mutants to be beneficial. And they're going to make it beneficial artificially. And that's what we're going to talk about next, is artificial selection and what exactly experimental evolution means. We're probably familiar with various forms of artificial selection. Here's an ancestral version of what we now know today as maize. A couple thousand years ago, the ancestral version of maize is teosinte. And teosinte wasn't a great food crop because it has these hard coverings over the kernels. And so it was really hard to get into the nutritious part of the corn. And over time, in central Mexico, the people that were living there started finding occasionally mutations that maybe made those fruit cups a little bit more accessible. And so what they would do is they would pick the plants that had the most accessible fruit cups, and they would selectively breed those. So they would raise those, and every once in a while you'd find a plant in the crop that had even more accessible fruit cups. And this is all due to random genetic mutations occurring as organisms reproduce. But over time, you keep selecting for the trait you're looking for, and eventually those farmers developed what we now know as corn or maize. So that's the process of artificial selection, and that's what these authors are going to do as well. They're going to create heat-shocked black mutants. They're going to take the greenest ones, because that's the trait they want. They want to make black mutants that will turn green to try to get a polyphenism. And they're going to basically throw out the rest of the Manduka offspring, the ones that aren't the greenest. And then you repeat that. So you take the greenest worms, wait until they have larvae, heat shock those larvae, see which of those larvae are the greenest, keep them, throw the other ones away, and then keep doing that for, I think they did 13 generations in this paper. And the idea here is to see if the authors can evolve a Manduka black mutant that will be green at higher temperatures, like its relative Manduka quinquemaculata. Just for fun, I thought I'd throw this idea in there because it's important for you to know about in Evo-Devo, and it's also relevant to this paper, that although this paper has nothing to do with crosses in particular, because this paper only studies that one line of black mutant Manduka, but in other forms of artificial selection, you'll see two different parents with two different phenotypes crossed to produce an offspring, in this case a hybrid, and sometimes those hybrids show what we call transgressive segregation or transgressive phenotypes. And what that means is that that individual is, in this case, larger than its two parents, and it also has different coloration, as it says in the legend there. And we see this pretty regularly in breeding programs, that sometimes hybrids between two different populations or two different species will have traits that are greater than, they transgress the range of traits in the parents. So here the parents are smaller, the hybrid is bigger. The parents have two different coloration schemes, the hybrids is different than either of the parents. It's not a blend of the two parents, it's something outside of the realm of what was found in the parental populations. And this also allows artificial selection on extreme traits. And so the reason I'm mentioning this is because this is kind of like what the authors of this Manduka paper are doing. But again, they're studying genetic variation within a species. They're not making hybrids, they're just heat shocking larvae and seeing how the different larvae, even in the same set of siblings. So imagine two parent Mandukas have a bunch of larvae, you heat shock them all, and as I mentioned before, different larvae will have different amounts of green coloration. And that's probably because of genetic variation mutations that are different between the two parents. And they come together in different combinations in each of those one individual offspring. And that is probably the basis for why some of the offspring turn greener and some less green and some just black like their parents. So we're not going to see transgressive segregation necessarily in what we're looking at in this paper, but I just wanted you to know about it, that in this case, this is another example from a paper on rice, looking at days to heading. That's a trait. It's related to flowering time for rice. And up here are shown the ranges of flowering times for two different parental lines of rice. So one parent had a flowering time that's very narrow, and the other parent also had a very narrow flowering time. But when you cross those two and look at F2, the grandchildren of the initial cross, the F2 generation, the difference, the breadth of this trait, flowering time, is way beyond the ranges that you see in either of the parents. You get rice that flower in as few as 70 days, as opposed to what either of the parents could do, even in the whole population of individuals from those parents came from. They have really narrow flowering times, but when you make a hybrid, all of a sudden, the range of phenotypes in this trait, flowering time, for example, expands greatly. This is a great example of transgressive segregation. So again, to recap again, this isn't the sort of transgressive segregation that we're going to see in the Manduka paper. This sort of thing has to do with setting crosses between very genetically different parents. In the Manduka case, the parents may be slightly genetically different, but the range and trait values we're going to look at also has to do with genetic variation, but probably like in this case, standing genetic variation between the parents. So when you see this sort of transgressive segregation, it suggests that the trait that we're looking at is complex, suggests that the trait involves the roles of many different genes, which is what complex means. A simple trait involves one gene, a complex trait involves many genes. And when you have crosses between individuals, genetically different individuals in the case of this rice cross, or genetically very similar individuals in the case of the Manduka sexta black cross, that range of traits that you get in the offspring can be an indication that there's some genetic variation that's segregating separately or independently into various offspring. And that's what can cause this range in trait values that we'll see in this paper. So finally, this is the last slide for this presentation. Let's go back to making some predictions about what might happen. So we're going to start with that Manduka sexta black mutant. We know that after heat shock, not after heat shock. Normally, at 20 degrees, all of the offspring will be black. At 28 degrees, all of the offspring will be black. But we know that with heat shock, if you heat shock those organisms, a red triangle or any sort of triangle can indicate heat, then they can become various shades of green. Light green to dark green, different parts of their body can be green. We'll see examples of this in the paper. So that is why the authors, again, are studying this black mutant, because they know it has the capacity, I shouldn't have said capacity because we talked about capacitors. It has the ability to become green, just like its relative that has a polyphenism. It just doesn't normally show this trait unless they're stressed. So the authors are going to stress the Manduka sexta black mutants, artificially select for the ones that are the most green, and see if they can develop polyphenism. So one thing is, if they get Manduka that are black when raised at 20 degrees, and if they can get them that are also same genotype, green at 28 degrees, then that phenotype is called genetic accommodation. So what that means is that new mutations have occurred during, maybe, or segregated into these organisms over the generations of experimental evolution that increase the sensitivity of the polyphenism to the environment. In other words, it suggests that what's happening as these experiments, the experimental evolution is proceeding, suggests that there could be some new mutations occurring or different rearrangements of alleles in the genome that give basically the ability of those black mutants the ability to respond to temperature like their relative does. So these two species, in other words, are very closely genetically related. But Punctum immaculata might have a mutation that allows it to respond to temperature, and that response might impact its pigmentation. And as I mentioned before, the reason that the authors choose Manduca sexta and Quinque maculata is because they're very closely related. So they're almost genetically identical. They're sister species. There are some genetic differences between them, but they're closely enough related that the authors hope that if they stress the black mutants, that they could overwhelm that capacitance of a canalized trait. Maybe it's a shallowly canalized trait. So they're thinking maybe they can stress the system just enough to get a little bit of that water out of the canal and change the black pigmentation to a green pigmentation like their close relative would do. So that's accommodation. So if the authors can get green consistently to develop anytime worms are raised at a consistent temperature of 28, I'm not talking about heat shock. They're just using heat shock to figure out which larvae can be green. And they purposefully then propagate those in the artificial selection scheme. They want to know if they can get a strain of Manduca sexta black mutants that will consistently be green without heat shock at 28 degrees. And if they can do that, if they can create a polyphenism, that is based on this concept of genetic accommodation. The other option, which the authors will tell us about, is what happens if you get experimental evolution to produce Manduca that are green at either temperature, like Manduca sexta. So they've taken the black mutant and they've experimentally evolved it to be green, but then it's just consistently green again. That is what, although this is kind of an artificial example because the black mutant is a mutant of a normal Manduca sexta, but if they can experimentally evolve green consistently, that is an example of genetic assimilation. And that is the canalization of a trait. So if they can get these strains that are consistently green, then that basically means they've dug a deeper trench. That's, in this case, there might be some mutations that have reinforced the ability of that trait to be consistent regardless of the environment. That's the main difference between accommodation and assimilation. Accommodation allows the polyphenism to exist, and assimilation is taking the transient, in this case, trait of a green Manduca from a black mutant and making it be the consistent trait regardless of temperature. And that's why it's called assimilation. It's taken a fleeting phenotype like green, but only if you're heat shocked and only if you're the one individual that's the greenest individual after the heat shock, and making it sort of the standard. That's why that's called assimilation. Thanks for watching, and I'm looking forward to discussing this paper with you.